A very warm greeting to all of you. Today is my great pleasure to share with you on our research entitled Comparative Analysis of the Physio-biochemical Responses of Kappaphycus alvarezii and Kappaphycus striatus to Elevated Temperature. The euchymatoid are red seaweed, which includes species from the genera of Kappaphycus and Euchyma, are cultivated in regions with a tropical or subtropical climate. Kappaphycus and Euchyma are cultivated mainly in the Coral Triangle, with the Indonesia, Philippines, and Malaysia ranked among the largest producers. Kappaphycus is the most prominent commercial source of carrageenan, which has a great economic value. As of today, there are 6 valid species of Kappaphycus. And among the 6 species of Kappaphycus, Kappaphycus alvarezii, Kappaphycus striatus, and Kappaphycus malaysianus are cultivated commercially in Malaysia. However, worldwide and commercially, the farmer mainly cultivated Kappaphycus alvarezii and Kappaphycus striatus. The farming of Kappaphycus has become a source of income and also being used for commercial production of carrageenan. It had also offered a means of livelihood to the local community and also to meet the increasing industrial demand for carrageenan. However, there are challenges being faced by this industry due to acute warming. In the absence of concrete measures to mitigate climate change, extreme events such as heat wave, typhoon, El Nino and La Nina have caused increase of seawater temperature. Studies have shown that due to abrupt changes in temperature, Kappaphycus are prone to ice disease. At the same time, ice disease are known also to have led to a significant decrease in seaweed production and decrease in carrageenan yield. It also reduced the quality and carrageenan viscosity and gel strength. The research objective of this study is to identify between Kappaphycus and alvarezii and Kappaphycus striatus, which species can withstand better with elevated temperature based on the physio-biochemical aspect, in which the growth, photosynthesis performance, pigment content, picocolloid yield, and quality, as well as ROS production were measured. This is the research methodology that was carried out. The samples were collected from Sempurna, which is the only commercial cultivated area in Malaysia, and subsequently the sample that were collected will be maintained and acclimatization from 5 to 7 days before we subjected for further analysis, and these are the parameters that were used for acclimatization. In addition to that, morphological as well as molecular identification were carried out to make sure that the correct species were used in this study. For the physiological and biochemical responses of Kappaphycus to elevated temperature, a total of 5 replicates each were carried out at temperature of 28, 32, 36, and 40, in which the growth response with a fresh weight measurement were carried out. Photosynthesis response were carried out using PEM photometer. Pigment contents on the chlorophyll A, carotenoid, and phycobiliproteins were carried out. Carrageenan quality tests as well as reactive oxygen assay were carried out. We hope that through this study, it will enhance our understanding on the growth, physiological, biochemical responses of Kappaphycus species toward elevated temperature, and we may identify which species of Kappaphycus can withstand high temperature, and this shall form as a basic for future work on long-term acclimatization to elevated temperature. Now let us move on to the result part. This is a graph showing the growth response of Kappaphycus alvarezii and Kappaphycus striatus. In general, the specific growth rate of Kappaphycus species decreased with increasing temperature above 28 degrees Celsius. Studies have shown that prolonged exposure to high temperature will lead to disruptive stress in the form of cellular and subcellular damage. These damages may lead to slow growth rate. In comparison between the two species, Kappaphycus alvarezii have better growth rate compared to Kappaphycus striatus, but there was no significant difference between the two species at temperature treatment of 28, 32, and 36 Celsius. Now let us look into regards about the external morphology of the growth response subject to various temperature. For Kappaphycus alvarezii, we can start to see whitening take place at the 10 day of 36 Celsius, and at 40 degrees Celsius, the whitening take place at much faster rate, which is on the second day. Okay, as regard for Kappaphycus striatus, at 36 Celsius degrees, there are some whitening take place, but not as much. Okay, but however, at 40 degree, we can start to see that the changes of color take place on the second day. Now let us look into the performance of the photosynthesis response when subjected to elevated temperature. For maximum quantum view, or generally known as FVFM, we can see that the FVFM value of Kappaphycus or Kappaphycus for both species are fluctuate in the range of 0.5 to 0.6 as 28 and 32 degrees Celsius. Thus, this shows the natural variability of FVFM and showing that they are at active photosynthesis rate. While at 36 degrees Celsius, the FVFM declined notably to 0.438 after 8 days of incubation, and further increase of temperature at 40 degrees negatively influenced the FVFM, lowering the reading drastically to 0.332 at day 2. The same features are shown in Kappaphycus striatus as well. The FVFM decreased as temperature increased. The FVFM value indicate that Kappaphycus striatus were much more healthier and have better photosynthetic performance compared to Kappaphycus alvarezii at 28 and 32 degrees Celsius. Studies have shown that high temperature stress reduce the photosynthetic parameter, indicating that a structural and functional disorder of photosynthesis apparatus and damage to the PS2 have occurred. Now let us look into regards about the light harvesting efficiency, or alpha. Alpha, which is the measure of the efficiency of carbon fixation per unit light, show that incubation at 36 and 40 notably inhibited the alpha, whereby the magnitude of inhibition increased with incubation time. In general, Kappaphycus striatus were much healthier and have better photosynthetic performance compared to Kappaphycus alvarezii at 28 and 32 degrees Celsius. As regards to the maximum relative electron transport rate, incubation at 36 and 40 notably inhibited the RETR max, whereby the magnitude of inhibition increased with incubation time. No major difference were observed between the two species at 28 and 32 degrees Celsius. Now let us look into the pigment content due to elevated temperature. Phycobiliprotein play a crucial role in capturing light energy and transferring it to the chlorophyll during photosynthesis. In this study, phycobiliprotein, which consisted of allophycocyanin, phycocyanin, and phycoerythrin content, was shown to be highest in strain when incubated at 28 degrees Celsius. Similar observations were found by Eretol et al. In 2014 on the brown and red variant of Kappaphycus alvarezii. Comparing the phycobiliprotein, Kappaphycus striatus has a higher content for allophycocyanin and phycocyanin at both 28 and 32 degrees Celsius in comparison to Kappaphycus alvarezii, but there was no significant difference in percentage mean inhibition between the temperature. As regards about chlorophyll content, as well as carotenoid of the two species were found to be highest at 28 degrees Celsius, and subsequently with higher temperature it showed a decrement. Incubation beyond the ambient temperature at 32, 36 and 40 degrees Celsius have led to reduction in pigment content after the temperature treatment. At temperature 28 and 32 degrees Celsius, Kappaphycus striatus has better chlorophyll A and carotenoid pigment content compared to Kappaphycus alvarezii, but there was no significant difference in percentage mean inhibition between both temperatures. Now let us look into the carrageenan quality due to elevated temperature. Based on current study, it shows that temperature can affect the yield and quality of carrageenan. As observed in our present study, which shows that seaweed incubated at 32, 36 and 40 degrees produce lower carrageenan yield compared to seaweed incubated at 28 degrees Celsius, which is the ambient temperature. In comparison the two species, Kappaphycus alvarezii have a better carrageenan and gel strength at 28 degrees Celsius, but increasing temperature to 32, Kappaphycus striatus have better performance in those qualities significantly. Gel viscosity between two species show no significant difference at both temperature. This shows that Kappaphycus striatus could perform better at elevated temperature in comparison to Kappaphycus alvarezii. Now let us look into reactive oxygen assay. As we are aware, the production of reactive oxygen is caused by inhibition of photosynthesis due to suboptimal condition, for example, high light, UV, high or low temperature. When seaweed are exposed to high temperature, photoinhibition occurs, which lead to accumulation of reduced electron acceptor and may increase the generation of reactive radicals such as reactive oxygen species, which can induce oxidative injury. The relative DCF fluorescence were reduced to 5,000 to 600 after first hour of incubation, indicate that the species are adapting to the temperature. In comparison the two species, Kappaphycus striatus have higher DCF fluorescence in comparison to Kappaphycus alvarezii for 48 hours. However, there was no significant difference in mean value between the two species at 4 temperature treatment. In conclusion, based on this study, we shows that Kappaphycus species was able to grow in wide range of temperature, but stress was observed at above 32, 36 and 40 degrees Celsius respectively. Temperature between 36 and 40 degrees can be considered as upper temperature limit for the study strain. According to the result, Kappaphycus striatus could withstand higher temperature for longer day, display a higher FeFM value, higher carrageenan yield and pigment content compared to Kappaphycus alvarezii. The results are expected to provide valuable information for farm management practice and to develop strategy to mitigate the effect of global warming. Okay. Here are the list of references being used for this presentation. And to end this talk, we'd like to acknowledge the funding agency and would like to thank you for your attention. Thank you.